And so we when we talk about arsenic and we talk about the strong association, we still do not have a really good example or good finalized mechanism of how this is carcinogenic. It's been hypothesized that there are genotoxic effects and DNA damage. It can alter enzymes and oxidative and create oxidative stress, which could promote carcinogenesis or interact with certain or repair pathways. The one I was talking to one of my colleagues recently, and we're in developing another study in environmental carcinogenesis, and we talk about almost the thought of evolution and how we've became from single cell organisms to what we are right now, and how we've developed and are the all living things have developed mechanisms to protect themselves from the environment or the exposures they are to exposed to. And the human body has a lot of pathways and mechanisms to correct them. Namely, a lot of our liver enzymes are actually meant to help combat a lot of these toxins. And so we hypothesize that there potentially not just arsenic, but other exposures and other environmental toxicants could just have our bodies have not developed the protective mechanism. So we don't really know for sure which way arsenic can. And it's been hypothesized it can interact with the sonic hedgehog gene and create mutations there at the basal level, and that's what creates carcinogenesis and bladder cancer. There was a study done in China about 3 years ago that showed it with several small study or a small sample size, but that was done all ex vivo. But again, there's multiple pathways and there's a lot of hypotheses, and I think that's going to be the next step of how we can, if we can figure out a mechanism of this carcinogenesis or potentially this co-mutagen, then we can figure out a way to target it or kind of combat it. Next slide. And now shifting over to disinfecting byproducts. Again, another environmental risk factor that's becoming more and more evident and or being studied more. And so DPPs, it's a little tongue twister. Form when chlorine is used for disinfecting drinking water and when they react with organic matter. The most common ones are trihalomethanes, so THMs, haloacetic acids, and nitrosamines. And so those are the three most common ones. We're going to focus a little bit on the trihalomethanes because those have been studied the most. Next slide. So again, what DPPs are the most concerning? Trihalmethanes are the most studied, so chloroform, bromoform, etc. Again, a lot of a key component of disinfecting byproducts. There are also some suggestions that nitrosamines and other nitrogenous compounds may be potent bladder cancer or carcinogens, but we still don't have a good grasp of all of the DPPs out there. Next slide. So what we from the epidemiologic literature, we do know that long-term exposure to these trihalomethanes is associated with increased bladder risk. Again, the risk is obviously again time-dependent, dose-dependent variables. There is an increased risk observed, especially at household exposure levels, which is above 49 micrograms per liter. There's been some conflicting data, but there's been some good data showing that some studies do indicate that there's a possible effect with showering and bathing, but I think the studies have been really conclusive in modern era. I think recently done at the NCI, showing that pool exposures really do not increase the risk. So it's thought to be due to the ingestion, but there are all potential mechanisms of how we can be exposed to these. Next slide. And again, Dr. Freeman, another excellent researcher over at the NIH, NCI, really focuses on this and really found that it's what's formed from chlorine and other disinfectants. There clearly showed that there's been elevated levels in these. And again, this was another two kind of case control studies. So it's a very large study done by big case controls with thousands of patients. Really how, again, this study really highlights, and a lot of the other ones we just talked about, really highlights how from the epidemiologic literature, we find these associations and potential causal or links to cancer development. And so Dr. Freeman did a beautiful job with this study, and I urge all of you guys to kind of take a look at this study because it really highlights a good way to do these big, large population studies. Next step, or next slide, excuse me. Um, in in that study, Dr. Freeman found that bladder cancer is positively associated with total THM or trihalomethane ingestion via drinking water in the top 5% of the distribution study. There were positive associations with higher than 46 micrograms, so the top 5% compared to the lowest quartile, which is less than 6. And again, that study that she did not support the association between swimming pool use and bladder cancer development. Next slide. And again, the mechanism of action again is all hypothetical at this point, but brominated THMs are metabolized and the reactive intermediates bind to DNA and they favor bladder epithelial cells and they can lead to mutagenesis, again, causing DNA damage and then triggering a sequence, and how a lot of cancers develop this repetitive sequence without being checked on and causing cancerous growth. Oxidative stress, again, potentially can, so THMs can create reactive oxygen species. Again, another way that we think of sometimes even patients in obesity and cancer development, especially in kidney cancer, causing an imbalance of cellular proteins. And it's hypothesized it can affect the NF-κB pathway, again, which is heavily involved in regulation of cancer development. And then we look at the epigenetic alterations as well, changing causing DNA methylation. And then another kind of aspect of cancer development, which I think we're learning more and more about, is endocrine disruption. And as we know, endocrine and hormonal functions is very important and potentially can be protective as well. And THMs may alter normal cell cycle and endocrine function, which kind of potentially could create bladder cancer as well. Next slide. And then shifting into nitrates. So nitrates is another environmental risk factor that we've been looking into as well. We, as the whole, meaning the whole bladder cancer community. And studies show that there is a positive association between higher nitrate concentrations in drinking water and an increased bladder risk. Again, potentially amongst patients and people exposed to long term. Again, so I think the key hallmark there is some sort of longevity, right? With this, like Dr. Silverman's kind of really showed that there's around a latency about 40 years. So we potentially are thinking about the same thing for these other risk factors. Dietary sources. I think that is one that it's another way that we can ingest and get nitrates. However, that's not been truly 100% proven. But high intake of nitrates and nitrates can be essentially processed meats have been linked to increased risk, but we don't really know the mechanism. Again, and then the mechanism that we think can lead to is just the endogenous formation of NOCs, which are probable carcinogens. And then another risk factor for nitrate development is risk lower intake of antioxidants, vitamin C, E, which can inhibit the formation of some of these. Next slide. So this was a study done looking at the nitrate from drinking water and diet and bladder cancer amongst postmenopausal women, specifically in the state of Iowa. And they looked at around 258 bladder cancer cases, including 130 among women. And they looked at their public water supplies for greater than 10 years. And they found that there was a significant association with the exposure, so greater than 4 years of that drinking water with greater than 5 milligrams per liter, compared to women who had 0 years of that comparable exposure. Again, another really good way to study this is the long-term or kind of longitudinal studies of looking patients and following them for several years and understanding their water supplies. And I think that's a brilliant way that our epidemiologists really learn about these, is that understanding getting these really good data from public water kind of municipalities and getting good data for that. So the study really did show that there is an association with dose, but also time. Next slide. And this again is another key study again done by Dr. Ward, and again looking at ingested nitrates and nitrites and bladder cancer. Again, and this is mainly through water supplies. And we believe that this is primarily from agricultural sources. So nitrogen and nitrates can be used and commonly found in fertilizers and manure and human waste, and we do know that they're often used in our agricultural communities. So in Dr. Ward's study, they found that the average drinking water nitrate concentration was above the 95th percentile compared to the lower ones in patients who developed bladder cancer. So again, another kind of concentration variable seen, and seen that there is an increased association with that as well. Next slide. Again, so if we will really look at the comparing the three that we highlighted today, we have arsenic, DPPs, and nitrates. So arsenic, I would say, we probably have the strongest evidence in terms of it being a carcinogen. Again, I think the mechanism is yet to be fully described. That's something that we're actually actively working on, Dr. Koutros and I. And really, this was actually commend Beacon for allowing this in patients from Beacon, as well as all the researchers, to create this almost a task force out of the think tank to really study this. And that's one of our research goals, is to figure out a mechanism for this. DPPs, again, I think there's moderate strength. I think there are good associations with bladder cancer, but we don't have a true understanding of a lot of them. Again, I think the trihalomethanes have been studied the best. And then nitrates as well. It's still emerging. I guess there's some uncertainty. I think there is clear an association. We're trying to understand that a little bit more as well. Next slide. And so the knowledge gaps in this are, I think I've alluded to this several times, is precise mechanisms. I think finding the mechanism of how these are either mutagens or co-mutagens, or how they disrupt our normal body's checkpoints, is going to be key in order to understanding bladder cancer development. I think we made a lot of progress, and again, namely due to our brilliant epidemiologists at the NCI, who really figured out and found dose associations. But I do believe that we can do better in this. And when is that ultimate trigger? We've hypothesized creating almost this calculation of a dose plus the time, and maybe that's the number that we need to figure out, and that's going to trigger someone to be an increased risk and maybe potential introduced screening. And again, going to that time, the time needed, right? I think we're learning that more, and I think again, Dr. Silman's study with the New England Water study was really pivotal in that. But I think for a lot of these other environmental risk factors, and name a lot that we did not cover today, that's something that's absolutely key. These three things: the mechanism, the dose, and the time. Next slide. So future directions, like I said, mechanistic studies, I think, are going to be key. And another thing is really understanding the exposome. So the exposome is, again, everything that we are exposed to. We're having we're having more developments in actually studying the exposome. So it's really hard to study exposures. And a lot of this are retrospective studies, or we're going back, or we're doing longitudinal studies, and we're following patients for X amount of years, but we're getting we're getting maybe good epidemiologic studies or information from that, saying their water quality, their duration of exposure, potentially the dose of what they're exposed to. But sometimes that's a very generalizable thing. Sometimes we don't know the individual. Also, we have not been able to really correlate that to, in terms of cancer, that individual's cancer. So what we've been trying to do here at Hopkins, and actually Dr. Koutros and I are doing a study together, which is currently in progress, is identifying a small cohort of patients and studying their hair, blood, and urine, which can potentially hold a little bit of more of what they've been exposed to, comparing that to their environmental exposures that were known through public data and epidemiologies, and looking directly at their tumor. And that kind of gets to the last bullet point for this is really study the interplay of the genetics of the individual, whether that's mainly their germline. And there's been a lot of germline whole wide association studies that have been coming out. And tumoral genetics, so the somatic kind of aspects of these tumors, and again, their environment and their exposome, and really creating a way to look at this from three different ways. And I think that would be the ultimate goal in environmental carcinogenesis. If we can really study this wholly, that would lead us to kind of more advancements in bladder cancer.